Maurício. Aumenta o deles um pouquinho.
Ok? Boa palestra. Good lecture, professor. Kimihito. Maurício.
Olá, boa tarde a todos e todas. Então, vamos fazer uma brevíssima apresentação da doutora Marilda e, em seguida, passar a palavra para ela dar continuidade aos trabalhos. A doutora Marilda Teixeira de Siqueira é graduada em Farmácia e Bioquímica pela Universidade Estadual de Londrina, tem mestrado em Biologia Parasitária pelo IOC e doutorado em Microbiologia pelo FRJ, além de ter realizado pós-doutorado na Inglaterra. Atualmente é pesquisadora titular da Fiocruz. Tem grande experiência com sarampo, rubéola, influenza e vírus essencial respiratório. É consultora da Organização Mundial de Saúde e membro de diversos comitês e grupos de trabalho do Ministério da Saúde e da OMS. Atualmente é membro de vários grupos internacionais sobre influenza, SARS-CoV-2 e outros ligados aos vírus respiratórios. Chefia o Laboratório de Vírus Respiratórios do sarampo do IOC, que é laboratório de referência nacional para influenza do Ministério da Saúde e da OMS, referência nacional para sarampo e rubéola pelo Ministério da Saúde, referência regional para sarampo e rubéola da OMS e para coronavírus em referência ao e também referência em coronavírus para as Américas. Desculpa a pequena confusão. Obrigado, Maria.
Eu não estou vendo ninguém aqui no Zoom. Só Maurício.
Boa tarde a todos. Eu queria primeiramente agradecer a presença de todos aqui nesse seminário que nós temos o prazer de receber aqui no Instituto Oswaldo Cruz, na Fundação Oswaldo Cruz, o doutor Ito. Ele é professor da Universidade de Hokkaido, no Japão, e é um cientista com uma experiência enorme em análises de dados, tendo contribuído os seus trabalhos científicos enormemente para diversas áreas, diversos vírus, entre os quais influenza, e mais recentemente para o SARS-CoV-2, nos quais os trabalhos publicados pelo doutor Ito e sua equipe contribuíram grandemente para o conhecimento epidemiológico, conhecimento da transmissibilidade das diversas variantes de SARS-CoV-2. Então, é um prazer muito grande nós recebermos nosso instituto, um pesquisador deste porte e que, nesses últimos dias, ministrou um curso de análise de dados para vários integrantes da rede genômica Fiocruz, patrocinado pela JICA, que é o Japan International Cooperation Agency, e do qual, então, vários de nós aqui presentes tivemos essa grande oportunidade de aprender nos últimos dias como melhorar as nossas análises e também discutir possibilidades de cooperação entre os diversos institutos e a universidade do qual o doutor Ito é professor. Então, muito obrigado pela presença do senhor aqui por nos dar esse seminário. Eu gostaria também de falar um pouco sobre essa cooperação da rede genômica Fiocruz com o Instituto Nacional de Doenças Infecciosas do Japão, aqui representado pelo doutor Itamura, e esse convênio que nós temos, então, com o apoio da JICA, entre esses dois institutos, Fiocruz e o NID, está nos proporcionando e vai nos proporcionar nos próximos 3 anos, e nós esperamos que depois, por mais tempo, a oportunidade de que vários de nós aqui, não só do Instituto, mas da Fiocruz, possamos, então, ir ao Japão, aprender metodologias, compartilhar experiências, compartilhar conhecimentos. Então, nós agradecemos imensamente à JICA, ao doutor Itamura, ao NID, essa oportunidade dessa cooperação entre o Brasil e o Japão, que já demonstrou muitas vezes responsável por progressos importantes em diversas áreas de conhecimento no nosso país, aqui na Fiocruz, através lá da década de 80, da transferência de tecnologia do Japão para a Fiocruz, para a produção da vacina de pólio e vacina anti-sarampo monovalente. Então, o nosso país, a Fiocruz, é extremamente agradecida a essa cooperação que nós temos com o governo japonês, esse financiamento da JICA, que tem nos proporcionado já há décadas avanços em diversas áreas de conhecimento e que vem contribuindo para melhorias significativas da saúde da nossa população. Então, nós agradecemos muito essa cooperação e agradecemos agora, particularmente, o doutor Ito por estar aqui e proporcionar para todos nós esse seminário. Muito obrigada e passo a palavra para o senhor.
Ok, boa tarde. Thank you very much for kind introduction. Yeah, so this is my first time to visit Brazil or even in South American countries, and then it's very wonderful things I have seen these days, and which we don't have in Japan, and then many things, everything is new to me. Okay, but I met many people who have academic view and scientific interest, which are very similar to me. It's a common interest and common mind about the science. Okay, that's very nice for me. Okay, so then my name is Kimihito Ito, and then today I'm going to talk about prediction of the trajectory of virus replacement. And then I'm from Hokkaido University, Japan, and then International Institute for Zoonosis Control, and then I'm a professor of Division of Bioinformatics. So let me introduce the Hokkaido University first. Okay, so then Hokkaido University, this is a Chinese characters for Hokkaido University. This is a Hokkaido University or something like this. And then this is another, it's a Japanese hiragana character, that's a Hokkaido Daigak. So then it's based on pronunciation, and this is based on meaning. And then this is north, okay, north sea, and then this is road, it's path or trajectory, and then this is large, and then this is study. Okay, so this is meaning of these Chinese character. And then we read this Hokkaido University. Okay, so it was found in 1876. as a Sapporo Agricultural College. So at that time, so we had a big transition from Japanese original shogunate, Edo shogunate period to Meiji era. So before that time we have shogun and the Edo period for 400 years. And then new government was established in around this time. And then new government want to westernize whole country. And then westernize everything, system and education and the agriculture and engineering, everything was brought by Western countries such as United States and the UK and France and Germany or such kind of countries. And then they put some agriculture school in Northern Island, Hokkaido. Okay, so Sapporo, and then in the city of Sapporo. So have you heard about Sapporo? It's a city, no name. So have you heard about Sapporo beer? Yes. Okay. So Sapporo is a beer. Is the first Japanese beer brewery. So this was also introduced introduced by the Japanese new government, and then they introduced technique from maybe Germany, I think, and then established Sapporo, first beer factory in Japan, in Sapporo, and then that is Sapporo beer. And then they want to westernize alcoholic drink as well. Okay, and then educational philosophy is of our university is frontier spirits, because Hokkaido is a northern end in Japan, and then it's almost Russia. Okay, and then it was just a forest, and then we want to Japanese government wanted to change this land to the new habitable place. Okay. Of course, there was some small number of indigenous people called Ain, and then but it was almost a forest because they lived together with nature. Okay, and then it is 100. It's a current picture of Sapporo city. It's modernized, and then you can see many buildings or something. But 150 years ago, it was just a forest, and then they built this university here. Okay, so you can see this city downtown area, and this is Sapporo station, and then we have a university campus. And then its second philosophy is a global perspectives. So because of this drastic change, we did not have any Japanese professor at that time. So we invite many professors from Western countries like the United States and England, and then all professor at the beginning were foreigners, non-Japanese people, and even president of university was from United States. Okay, so then we started as some kind of the environment with international. It's international environment from the very beginning of this established of university, and all-round education, and then practical learning. So because this agriculture, its area of agriculture is practical. Okay, so it's very, it's a study of the agriculture should be very directly transferred to the other people, farmers or something. It's so we think that it's science need to be connected to some practical implementation and which help society. And then these are four education philosophies of the Hokkaido University. And then we have 11,000 undergraduate students and 6,000 graduate students. Okay. And then this is a picture of the institute where I belong to. And then it was established as a research center for zoonosis control in 2005. Okay, so the purpose of this establishment of this was so it's having researchers from medical science and veterinary science and other discipline to conduct new research area of zoonosis control. Because as you know, so almost all emerging disease are zoonotic. Okay, so virus cannot emerge without transmission from some animals. Okay, so human virus, we have human viruses, of course, this is, which was circulating, which has been circulating for a long time. So that is human infectious diseases. But emerging infectious disease occurs when a virus came from other place to human society. The other place is the other animals, for instance, wildlife or livestock, and such as things. Okay, so no one developed new virus and spread. So that is not the true story. So always emerging disease come from other animals, wildlife and livestock. So disease of livestock or animals is handled by veterinarians, right? So veterinary science is responsible for the infections in animals. And then medicine is responsible for human infectious diseases. But zoonosis is no responsible academic disciplines. Okay, so then Hokkaido University thought we need to have some new institute which specifically deal with zoonosis. So SARS-CoV-2 zoonosis as well, and then influenza is also zoonosis which transmitted from pigs to human population. So and other monkeypox, mpox is also zoonosis, and then such new virus, new infectious disease are almost 100% zoonosis. And then so we have designated as a WHO collaborating center for zoonosis control in 2011. Okay, so let me introduce myself. So my name is Kimihito Ito. So this is character for me. So this is, so in Japan we write our name from the last name. It's a surname first, and then last name the first name comes last. So then surname and a given name. So surname is Ito, and this means some priest or something. I'm not, which is far from my job. But, and then this character is for the wisteria. It's a kind of flower. Okay, and then so this one is so this was about Ito, and then Kimihito is this part, Kimi and Hito, and then Hito, this means a person. You can look like it's a person is walking. This is person, Chinese kind of character of person. Okay, and then this one means public or something, or fair or public or such kind of meaning. So it's a public person. Okay, so and then let me introduce myself, background of myself. So I graduated school of Hokkaido University, and the graduate school of engineering in 1999. Okay, and then I got PhD in 1999, and then in the field of the information science, and then machine learning, and at that time I was doing machine learning and data mining or something like this. So very basic research about this one. And then after the graduations, I became some assistant professor at the Graduate School of Information Science and Technology, and then I was working there up to 2005. So when we established Research Center for Zoonosis Control, and then after that I moved to this university and became an associate professor of this Zoonosis Control Research Center. And then from that time, I applied machine learning approach for the prediction of evolution of influenza A viruses. So it was almost 19 years ago. And then recently, our institute changed the name from Research Center for Zoonosis Control to International Institute for Zoonosis Control. Okay. Okay, so pathogens, as I have told, and the pathogens originated from wild animals or livestock, cause infectious, emerging infectious diseases. Okay, and then so we have many pathogens or viruses or bacteria in wild animal or livestock population, and then when it transmits to the human populations, and if they adapt to human, and then if they are transmissible enough for the human-to-human transmission, then a pandemic starts, because human population haven't faced this viruses, and then we don't have any immunity to these viruses, and then pandemic occurs, as we have seen in the SARS-CoV-2 pandemic. Okay, and then after the pandemic, if this there are two cases, two scenarios. One is after when many people have have been infected by these viruses, and then we have immunity to viruses. And then population immunity, due to the population immunity, new virus cannot survive and they extinct. So this is one story, but it's one scenario. But in some cases that might be possibility, but many cases we have seen that cause epidemic, and then we have seasonal epidemic of such a pandemic viruses. So influenza H1N1 was also pandemic viruses, but after introduction, they still continue to circulate in human population. And then also SARS-CoV-2 also. So most of people have infected by SARS-CoV-2 viruses, but they still have ability to circulate because these are because of their evolution. So, okay, and then in this seminar, I'm focused on the evolution or selection, natural selection of the viruses, and then how to predict this selection process. Okay. Okay, so let me introduce a little bit about the viruses. What is viruses? Okay, so here we have electric microscope picture, and then electron microscope picture. So this left-hand side is influenza viruses, and the right-hand side is SARS-CoV-2. And then both has some spike proteins around here, which is needed to attach the cell surface. And then inside, they have RNAs, which is their genomic information is written, kept in these RNAs. One is a negative strand and the other is a positive strand, but it's basically we have they have genetic information, and then some virus has genetic information in DNA, and then some other RNA or something like that. But it's common thing about virus is that they cannot replicate them by themselves, right? So even if you get some many material which needs for the new viruses, then they cannot replicate. They need a live cell or living cell to replicate. So then basically after the attachment, and then they are transported to the inside of the cell, and they release their RNA to the inside the cell. Okay, and then in the cell is always reading our messenger RNA, and then making proteins, which is necessary to our activities. So cell needs some many proteins, and then this cell keep producing proteins by reading messenger RNA encoded in our transcribed from our genome sequence, and then protein we need is normally produced. But in the case of the viruses, virus release some their genome, and then hitchhike this system, and then host starts to produce proteins which is necessary for the viruses. And then if every proteins they need, and they they assembled at the surface of the cell. But the important thing that our cell does not replicate the viral RNA. So they have their own polymerase which replicate the viral RNA. And then we have something, and then we get this. We get everything which was inside in original viruses. All proteins and the polymerase, and then RNA, and then it's bud from the cell surface. So that is a replication of viruses. So let me introduce that something similar to the viruses. Have you received this kind of chain letter? Like this is called the letter of happiness. So it is no, we don't have this anymore, but it's a kind of chain mail. So we receive chain mail now by the smartphones or iPhone or Android. We have, or email, we have receive some chain mail. So if you receive this email, please send this email to others. So that is a kind of chain mail. So I have received some email that, so some dogs are kept in public health authorities, and then they want to know, they want to find some new owner for these dogs. 100 dogs, and then if you know someone who likes to have dog, then please distribute this email to them, or something like this. And I distribute it, but it was chain mail. Okay, such kind of thing is chain mail. So then, but 50 years ago. We don't have emails, of course. And then at that time, this kind of chain letter came with the physical letter. So this is typed by typewriters. They said that if you receive this email, please distribute the copy of this email to others. So if you do this within 1 week, you have very good happiness. But if you don't do this, and or tell about this mail to others, then if you don't make a copy of this one, then you have very bad luck. So and then this is it's written some scary things. If this person killed by something because they don't make a copy, or it's very scary thing, and then very happy thing, and he received $40,000 and such kind of thing. Okay, so this is information passed from readers to readers, right? Okay, so they have ability to make a copy of themselves. They cannot copy by themselves, but they use the letter used to humans to make a copy of themselves. Okay, so now this electric version don't mutate, so it's not very strong. But this one was typed by typewriters, and they sometimes people have a mistake, grammatical errors or something, and then sometimes they add more some good or it's some scary thing or something. So if the mutation is bad thing for so it's for instance that misspell or grammatical errors, or lack of important sentence, then these letters have lower probability of surviving. Okay? And then if they have some good phrase, they obtain good phrase of replicating, then their probability of survival, or the probability of the readers to make a copy, increase. Okay? So then during the circulations, only good letters survives. So after the months of the circulation, the letter becomes like this, very sophisticated letters. So letter does not have any, doesn't know about any English grammars or something, but it is the letter with good phrase can survive. Okay, so this is, and then after the circulation, letter becomes better and better. Okay, so that is a mechanism of evolutions. So they don't need anything. They just need a human to understand this letter. Okay, so letter with trustful phrase had higher probability of replication than those with untrustful information. So obtain a and at last obtain a sophisticated message for readers to copy. Okay, so this is basically virus is the same. Okay, if they virus when the virus replicate, the polymerase introduce some errors. So this error, if this error is good for the further transmission, then they are selected. And then if the mutations is not good for the further circulation, then they will extinct. So as a result, we have better and better genomic sequences, and the transmissibility becomes better, higher and higher and higher, which we are seeing. So for the SARS-CoV-2 or otherwise, other things. So basically, this is it's mechanism in nature, but a human or ourselves is evolved like this. Okay, so this is basic mechanism. Natural selection is basic mechanism in we have in for thousands of the years. Okay, okay. So this is some things about chain letters. Okay, so if you are interested in about the letter, there is some journal article about that one. Okay, so then okay, so natural selection of virus is something like this. Replications induce a copy errors in RNA or DNA. So large variety of descendants are made. Okay, so and then viruses about adapting the environment are selected. So then, and then this is repeated and repeated and repeated, and then finally, and then virus evolves. Okay, so it's they are not mutating by themselves, right? Just they are just we are or they are just selected by environment, like letter. They don't have any brain. They don't have any strategy. This is just the nature of evolution. Okay, so let's look at the nucleotide sequences. So this is I'm using nucleotide sequences of influenza viruses. So this one is HA sequences of the H2N2 viruses in 1968, and then this is sequences of 3 years later after pandemic, Hong Kong pandemic, and then you can see yellow parts changed, and then these are 40 years later, we have many sequence changes. Maybe some of them are increased transmissibility or escape the immune obtained by the previous infections or something. Or one is good for the immune escaped, and there's something is good for some mutation may be good for the adaptation of the pig virus to the human population. And then some are just random mutations which does not affect viral fitness or something like this. Okay. Okay, so then, so the information I showed, it was genetic information. But we also have another type of data. So epidemiology and the genetics is two different disciplines. Okay, so researchers in epidemiological analysis are interested in the number of infections like this. This is a number of infections in the SARS-CoV-2 virus pandemic, and we have some waves or something like this. And then their interest is to estimate basic reproduction number or effective reproduction number. And they use some mathematical model such as SIR model or renewal model. Okay. On the other hand, there is another group of people who are analyzing pandemic. They are genetics. So genetic analysis basically collects nucleotide sequences from the patient and then draw some phylogenetic analysis, and then using some population genetics, and then analyze the presence of a natural selection or something like this. So these are basically different scientific field, right? But the phenomena we are handling is the same thing. Okay, one phenomena, which is SARS-CoV-2 pandemic, but different discipline use different approach. But we think that we need to analyze the both. Okay, so we need to combine these two different areas because it's more powerful. Okay, so then from the epidemiological point of view, by using genetic information, they can get some more understanding. Okay, and then from genetic analysis side, if they have some such kind of epidemiological data, we can use some that one we should use. So then our purpose is that to merge these different areas, to conduct new study by merging these two different areas. So let's look at the data first. So we have enough data. So in the case of influenza viruses, weekly number of infections were reported. And then sequences of 500,000 strains are stored in the GSED. And then including 100,000 from H1N1 pandemic strains. So we accumulated 100,000 sequences from 2009. Maybe it's 15 years, and then 100,000. Okay, let's look at SARS-CoV-2. In the SARS-CoV-2, daily number of infections and deaths were reported by WHO and countries. Well, nowadays, depending on the countries, they switch to the weekly report or something. But at least during the pandemic period, WHO report these numbers every day. And then sequences of SARS-CoV-2 is now about 16 million. So it's just the result of 4 years. 4 years, 16 million. And then in that case, 15 years and 100,000. So you can see how big data is accumulated for SARS-CoV-2 in both terms. Okay, epidemiological data and the sequence data. Okay, so let me introduce our first work with an famous epidemiologist in Japan, who is Nishiura in Kyoto University. I've known him for 15 years, and then we had some joint. We received a grant from the government, and then we conducted some joint research about this combination of this genetic information and then epidemiological information by the government. But at that time, we are handling with influenza viruses, and then we did not have good. We developed some method, but we did not have good chance to apply it because of some reasons. But at the end of the project, we had SARS-CoV-2 pandemic, and then we applied our method to the SARS-CoV-2, and then which was very successful. Okay, so let me okay, let's think about situation. Let's think back. To the situation in June 2021. So Delta variant showed high transmissibility in India and the United Kingdom. So Tokyo Olympic Games are scheduled to start from July 23 in 2021. And the replacement of Alpha by Delta was posing a public health threat in Japan. But we did not have enough information about Delta. So then important questions of June 2021 was how transmissible Delta is compared to Alpha. And then when Alpha will be replaced by Delta. Because next month we have Olympic Games. And then Delta is coming. And then we wanted to know when Delta replaces the Alpha. Okay, so then we did some calculation of relative reproduction number, and then which needs the relative reproduction number is that ratio of effective reproduction number. Let me introduce effective reproduction number first. So effective reproduction number is average number of people, uninfected people at time t could be expected to infect. So this is the formula to calculate effective reproduction number. So basically, I'm not going into this detail about this formula, but basically the numerator is number of new cases at now, okay, today. The denominator is a number of infections in the previous generations. And then that one denominator can be calculated by the generation time distribution multiplied by the number of cases in the past. Okay, so this is, if you want to know this more, then just please read my paper, or please ask the organizer of this seminar, because we had a workshop in the last 3 days about this. Okay, so then a relative reproduction number is defined as a ratio of effective instantaneous reproduction number, which I shown in the previous slide. So this is suppose this is relative effective reproduction number of newly introduced variant divided by effective reproduction number of existing baseline strain, and then this one is called Ki. So we have some strong assumption of that Ki is constant over time. So it means that if Ki is, for instance, 2.0, then it means that new variant is 2.0 times more transmissible than previous variants. That is a definition of relative reproduction numbers. Okay, so then our main contribution is that to found this formula to predict or to model the virus replacement. So virus replacement by Q is a frequency of the new variant can be modeled by this formula. So which Ki is a relative reproduction number, and the Gi is a generation time distribution. These are frequency in the past or something like this. So there's no need to know the effective reproduction number itself, and then no need for the information of number of infections. So just a very simple one. So past frequency and the generation time, and then relative reproduction number, then we can generate we can model the replacement trajectory by this formula. And then this is the results of the application of that formula to the delta alpha, about delta prediction. Okay, so then this is the data from Japan. Then at the end of the 2020, we have some Wuhan originated ordinary strain of this is Wuhan type strain, which is yellow. And then we had some Japanese original mutant called R1. R1 strains came, and then it is original Wuhan strains were replaced by the R1 in Japan. But at the same time, alpha variant was introduced in Japan, and then they start to grow around here, and then replaced R1 strain completely around May or something like that. And then we can see very small number of delta like this. This is which is representing in a green plus. Okay, so from this small increase, we fit the model to this observations. Then we get we estimates of the relative reproduction number, and then result was like this. R1 strains is 1.25 times more transmissible than original Wuhan strains. And then alpha variant was 1.44 more transmissible than original Wuhan strain. And the Delta was 1.95 times more transmissible than original original Wuhan strain. So, and then, so it is predicted that relative reproduction number in the virus population will increase by the replacement of the Delta, replacement of the Alpha by Delta. Okay, and then this was the result was reported to the Japanese government through the ministry administration. It's SARS-CoV-2 advisory board of Ministry of Health and Health and Welfare. Okay, but that was just a prediction. So let's check that prediction was correct or not. Okay, so this was a prediction we did in June 23rd, which was reported by to the government. And then these are the trajectory of the observations after the report. Okay, so you can see that these frequency trajectory is perfectly match the predictions. Okay. Okay, so that is the first result. And the second result I'm going to show you is about Omicron. So situation in the, this is a situation in the December 2021. The Omicron variant showed high transmissibility in South Africa. And the replacement of Delta by Omicron was posing public health threat worldwide. So important question is similar, but just players are different. How transmissible Omicron is compared to Delta, and when Delta will be replaced by Omicron. Okay, so and then we analyzed the data from Denmark, because Denmark had huge number of sequences. They basically sequence all specimens they collected and submit to GSET, and they have very good quality of the data. And then you can see this very beautiful curve of increasing of the Delta and increasing of Omicron and the decreasing of Delta. Using this curve, we estimated that that Omicron is 3.19 times more transmissible than Delta. It's very transmissible. And then we published this one in end of 2020. Maybe, yeah, it's in the very early stage of the Omicron replacement. So this result was introduced by CDC, and then CNN also reported that about our result, and then it is used in the risk communication or something like that. Okay, so and then let me show you the result of prediction. Okay, so result of prediction is almost good until here, but after 50% we have some mismatch between prediction and observations. So we did not know about the reason for this. So we wanted to try to we tried to clarify the reason for this one. So you can see that asymmetry in the replacement. This one is very around here. Around here, its replacement is very quick, right? But after 50%, replacement slowed down. So we wanted to know the reason, and then we solved this one by assuming the generation time of Omicron is shorter than Delta. If we assume that, if we allow the difference of the generation time, then it completely matches the actual observation. And then we conclude that generation time of Omicron is about half of Delta. So we don't know this is correct or not, but it is already known that Omicron is generation time of Omicron is shorter than that of Delta in other epidemiological data. Okay, so this is second one. Okay, so then third one, it's about how accurate prediction is. Okay, so we did some kind of retrospective prediction test. So using alpha delta transition in UK. Okay, so you can see that transition from alpha and delta here, and then you can see that very beautiful curve of the transitions. So we use UK data because they have good samples of the very large number of nucleotide sequences, and maybe it's random sampling from the population or something. I don't know, but anyway, it's very beautiful curve. Okay, so prediction was made using relative reproduction number when the relative frequency of delta was 5%, so and 10% and 50% and up to 90%. So it's basically so this first line is the 5% line. So the day delta exceeded the day delta reached 5%. Okay, and then we hide all observations after this day and then predict future and then compare the results. Okay, and then so next line is that the day delta reached 10% and hide all data after this date and predict future using the data before this date. So this is the result of the prediction when we used the data before the day delta reached 5%. So when delta was 5%, the prediction was like this. So then it is this is red line indicates the predictions, and then black line indicates 95% confidence intervals. So then red circles is future data after the prediction, and then you can see some it's almost within the 95% confidence interval, but we have some discrepancy between prediction and then the actual data. So then maybe 5% is too early. And if 10%, it's a little bit improved like this, and then, but still we have some discrepancy. But 15%, we have almost perfect match. So of course, 20% perfect and 25% and perfect 30% and so on. It's everything after 15% is okay. So then these are the error of the predictions. This is the actual date delta reached 90%. So this is a date of the predictions. Okay, and these are predicted date when delta reached 90%. This is something overestimated because delta reached very early, and then this is also this is 5%, 10%, and this is prediction at 15%. So our conclusion: once new variants relative frequency reached 15%, the date when the relative frequency of the new variant would reach 90% was predicted with maximum absolute prediction error of 3 days. Just 3 days error. It's very, we can do the very accurate predictions if we have such kind of very good quality data. Okay, but we need to wait until it becomes 15%. Okay, and finally, the final result I would like to share is joint work with Fiocruz and Hokkaido, our Hokkaido University. So this is published by the Nature Communications. The first author is Igor-san, and then corresponding authors are. Last author is Felipe-san, and then corresponding authors are Gonzalo-san and Felipe-san. Oh, okay, these are maybe people you know very well. Okay, and then okay, so then this is like result was like this. So I was responsible only for this figure. Okay, every other thing is done by Fiocruz team. So then I just want to introduce only this part. It has many other important analysis like phyrodynamics or something like that. Okay, so this is just one of the results in that paper, which I'm responsible. So relative reproduction number of gamma and Omicron BA1 with respect to Delta was estimated to the 0.76 and 3.25. Okay, so it means that gamma is 0.76 0.67 times more transmissible than Delta. So it's less transmissible than compared to Delta. And then on the other hand, the Omicron BA1 is 3.25 more transmissible than Delta. And then you can see this quick replacement. And then it starts from so at that time, at the beginning, the gamma was dominant, and then replaced by the Delta, and then relative transmissibility a little bit increased, and then by the replacement it increased up to 3.25. Okay, so that is the beginning of the collaboration between Fiocruz and then Hokkaido University or me. Okay, and then that was very good. I'm very happy that it was published in a very good journal. Okay. Okay, and then I would like to thank very much to all of you, of these authors, because it's really nice result. Okay, so then this is summary of this my talk. So then it is not possible to stop the replacement of virus. Don't try to do this. It's impossible. So it's a good one replace the others, bad one. So we cannot stop this process. This is just nature of the evolution. So it's every organism is living like this. We cannot stop the evolution. So some future trajectory can be predicted, and then accurate prediction were expected after the new variant reached around 15%. So, but 15% is about just 1 month. So we only have 1 month from. So the public health policymakers may have only one short time to prepare control measures for the increase in the viral transmissibility, and a quick decision-making process is needed to take advantage of the prediction. So we can predict, right? Like 1 month before the Olympic Games, okay? But it is so decision makers need to prepare very new strategy in a very new quick way. So in that case, in the case of the Delta introduction, so Japanese government decided to have Olympic Games without audience, only athletes and the judge and some staff can allow to enter the stadium or place they are competing each other. So they decide no observatory, and then maybe I don't know they use our prediction or not, but it's I think anyway. So they need policymakers need to decide new strategy in very short time. So short time prediction is possible, but it's long prediction is very difficult. Okay, so that is my seminar. Okay, thank you very much for attendance. Okay.
Muito obrigada. Thank you very much. Arigato. Como é que fala? Yeah, thank you. Eu esqueci a segunda parte da frase. Bom, muito obrigado. Foi um excelente seminário, super interessante. Eu, pessoalmente, gostei de tudo. Gostei muito da gente ver a história da tua universidade, entender um pouquinho dessa cultura em termos de língua, de linguística japonesa maravilhosa. E também achei muito interessante a história da carta. A gente não pensa muito nisso, né? Mas é muito bacana mesmo a gente entender, ter modelos diferentes daquilo que a gente pensa sempre, para entender essa questão de evolução, de quem permanece. Muito bacana, muito bom. Muito obrigado por todo o seminário. Eu gostaria, então, de saber se alguém tem alguma pergunta, algum comentário. Tem alguma coisa? Você tem. Vamos lá. Gonzalo, por favor.
Okay.
Yes, okay.
Thanks very much, Dr. Ito, for all these days with us, and for the workshop, for the seminar of today. One of your conclusion is that it is not possible to stop the replacement of variants. But what we have seen in the last 2 years, maybe, after the emergence of the BA1 variant, is that new lineage replacement will not cause a very large upsurge of severe case.
Okay.
Severe case. So do you think that maybe we need to couple the ability to predict which variant will become dominant with some measure of the ability of that variant to evade immune response and cause a severe infection?
Okay, so that it's a replacement and then number of infections, a number of cases or epicurve is not, it's not necessarily correlated. So even if we have so if we have a higher the variant with a higher transmissibility, then always replace the others. So it does not. Sometimes we have new outbreak because of the replacement, but sometimes we don't have. Even if we have replacement, but for instance, if we have very small difference, but replacement will. Let's suppose some two variants which have similar transmissibility. Relative reproduction number is very similar, but it's one is more transmissible than others. Then we have the replacement of the low variants with lower transmissibility or lower reproduction number is replaced by that with higher transmissibility. So this process cannot stop. So it is no method to stop this one. But sometimes we have the large outbreak by the replacement, but if the two variants have similar reproduction number, then maybe we don't have large outbreak. Just a number of cases goes down, or goes down, or just flat, or the similar situations. Okay, so for instance, when we have reproduction number below 1, for instance, let's say 0.9, then number of new cases decline like this. Okay, and then if we have two variants, one is which one is let's say okay, so let's say that one is a little bit more transmissible than others. In that case, both variants, reproduction number of both variants are still smaller than 1. Okay, so it is, for instance, one reproduction number is 0.9, and the reproduction of others is 0.8. But if you compare 0.9 and 0.8, so 0.9, it means it's more transmissible. Then this is a relative reproduction number, it's 9/8, which is 1 point something something. Then replacement happens. But reproduction number is 0.9 and 0.8. The epidemic is still declining and declining and declining. So that is the case. We don't have a large outbreak, even if we had a replacement. Okay.
Okay, but yeah, no, but I mean not only the ability of the new variant to increase or not the number, but about the severity of case. Because if the number increase, but most cases are asymptomatic or mid, that is not a big problem for the public health system.
Okay.
It's something that happened with the viral evolution, but will not have very bad consequence for the overload of the public health. So I think that we also need the ability to predict the
Okay,
virulence, the pathogenicity of the new.
Okay,
Viral.
yeah, right. That's very important question. Okay, so then at this one, alpha delta replacement. Okay, or this one is also alpha delta replacement. So if you know that how severe delta is compared to alpha, then we can use that information combined with this one. So we know we can predict the proportion of the delta infection in the future, and then we combined the severity of delta. Okay, so then we can know something about the proportion of severe cases, but still we have some need something to predict the cases, or to predict number of cases in the future. So we can predict the replacement, but we cannot predict the number of cases, because number of cases heavily depends on the status of the communities, social distancing, or it's lockdown, or vaccinations, or other so drug use, or I don't know. So it's such kind of factor is related to the number of new cases. But the replacement itself is not affected by these things. Okay, so we can predict the replacement, and then we cannot stop the replacement.
Obrigado.
It's just a quick question, because it's something we escaped our discussions in the workshop. For the public healthcare system, for the decision makers and stakeholders of the healthcare system, it's very important to combine a model that can understand the rate of replacement, but which particular lineages variants may cause an increase in number of cases, but more likely, Gonzalo said, we cause more severe symptoms. We had only proper 2 waves that cause severe cases, the first one, and the second with VOC gamma. And we know the relative reproduction number may become constant, but the actual reproductive number is not constant number, and when it goes below 1, the number of cases goes down. And so for us, we wanted to think about if there is any possibility incorporating your model to another one that could predict a spike of cases, because we knew certain strains would be replaced, like gamma would be replaced by delta, but didn't cause a wave. Omicron replaced delta and caused a huge wave. And then BA2 replaced BA1, but didn't cause a wave. And then BA4, BA5 caused a wave, had a wave replacing BA2. And then there was another one in November and December 2022 during the World Cup. And then the BQ1, BE9 replaced the others, BA5, BA4, and caused a wave. And then XBB didn't really cause a wave, but JE1 caused a wave only a few states. And that kind of thing that we wanted to have more or less in prediction. Replacement is really nice, but a spike of cases and severe cases would be even better.
Okay, so we need to decompose the advantage of the into the advantage from the immunity, or advantage from the transmission. So then, at the beginning, nobody have immunity. Okay, so at the beginning of pandemic, nobody had immunity to this. So then, we don't need it to. We didn't need to incorporate its immunity, but it's a current situation. So we need to take into account the immunity to modeling. But it's some future work. I have no formula yet, but it's very good. It's some right way to go. Thank you for the comment.
Mais alguma pergunta, Igor?
Thank you for your presentation. I was wondering, is there any new feature that you plan to implement in new versions of your program, of the RailRe program?
Not yet. Not yet. I have no idea. But I knew that is important. I knew, but I have no good idea.
Ok, mais algum comentário, alguma pergunta? Não, então eu gostaria mais uma vez de agradecer ao Dr. Ito essa oportunidade de discutirmos com ele. Eu vou fazer só um comentário, aí eu gostaria até que o Dr. Ito confirmasse ou não se eu estou certa ou errada. Para nós termos assim, uma ideia, eu já comentei isso com um grupo do nosso laboratório, né? O Japão, por exemplo, não fez lockdown na pandemia, não esse lockdown que nós fizemos aqui no Brasil, que a maior parte dos países do mundo fez. Eles fizeram alguns lockdowns pontuais e muito baseado nesses estudos, em resultados que eles estavam tendo de uma vigilância que unia não só uma vigilância epidemiológica clássica, mas outros dados também que permitiram, então, que eles fizessem estratégias de controle e que foram muito bem sucedidas. Né? O Japão tem 120 milhões de habitantes, nós aqui no Brasil temos 202, então eles têm aí 60% a nossa população, e eles tiveram 30 mil mortes, mais ou menos, e nós sabemos quantas nós tivemos. Então, a gente, é claro que eles são um povo que tem mais facilidade de entender o que as autoridades sanitárias estão pedindo em determinados momentos, né? Enfim, eles têm um controle melhor, enfim, tudo mais. Não vamos falar mal dos brasileiros, né? Só vamos falar mas naturalmente que eles uniram uma série de estratégias e foram muito bem sucedidos, sem a necessidade daquele lockdown que a gente teve e que a maioria dos países do mundo tiveram. Então, nós, como Fiocruz, junto com muitas outras organizações internacionais, OMS, OPAS e muitas outras, estamos trabalhando muito, né, na questão de preparação para uma próxima pandemia, para que a gente, numa próxima pandemia, tenhamos um enfrentamento melhor, menos impactante em termos de mortalidade econômica e também a questão social, a questão de saúde mental, tudo que impactou. Então, é uma oportunidade muito legal que a gente está tendo agora de poder, então, naquilo que a gente pode, como laboratório, como instituição, melhorar em termos de conhecimento que nós podemos, então, aplicar não só no nosso dia a dia, mas também na nossa preparação futura. Então, muito obrigado. Alguém tem... Tem o chat. Acho que tem uma pergunta no chat. Você consegue ver para mim, Gonzalo? Tem... Só elogios. Ah, ótimo. Onde é que estão? São comentários, tá bom. Então, alguns comentários no chat. Então, muito obrigado a todos. Obrigado, doutor Ito. Obrigado, doutor Itamura, mais uma vez, Mariko, Tominaga, por toda a parceria que nós esperamos que seja cada vez mais frutífera entre as nossas instituições. Obrigado, boa tarde, bom final de semana.